A pleasant morning to all. On behalf of my advisor and my co-authors, I'm honored to present our research to you. World Health Organization reported that cancer is the leading cause of death worldwide, accounting for 7.6 million deaths in 2008. It was projected to continue rising with an estimated 13.1 million deaths by 2030. Therefore, there is a need for the development of new chemopreventive agents. May I present to you our research entitled Ulvalactoca, a potential treatment for leukemia. Ulvalactoca is a green seaweed belonging to the family Ulvaceae. It may be found attached to rocks and shells by a holdfast, but commonly found free-floating. It contains large amount of polysaccharides, which constitute 38 to 50% of the dry matter. A particular polysaccharide that can be extracted from Ulvalactoca is called ulvan. Ulvalactoca may be found in Philippines in places such as Batanes, Palawan, Zamboanga, and almost all over the country. This suggests that Ulvalactoca is widely distributed here in our country. It contains various health benefits such as antimicrobial, antioxidant, anti-malaria, anti-hypertensive, anti-diabetic, anti-inflammatory, antiviral, anti-helminthic, and immunosuppressive property. The objectives of our study is to characterize Ulvalactoca, to extract and purify ulvan from Ulvalactoca, to analyze the anti-leukemic property of ulvan against leukemic blood. The significance of our study is to establish basis for a possible cure for leukemia that is low cost and have minimum or no side effects. Methodology. Collection of the sample. The sample was obtained near the Gulf Beach Resort, Zamboanga City, last July 2012. It was transported to the laboratory with ice in an ice box. Then, it was washed with distilled water thoroughly to remove extraneous matter and to avoid further microbial contamination. 100 grams of the sample was submitted to the National Museum of the Philippines for authentication. 70 grams of the sample was soaked in distilled water, and then the suspending water was removed after sanitation. Then 3 ml of potassium acetate was added to the tissue extract. Then equal amounts of ethyl alcohol was also added to precipitate the ulvan. The precipitate was placed in a vacuum desiccator for drying and hardening for 24 hours. Cell preparation and isolation. On the other hand, the leukemic blood obtained from the hospital was concentrated using histopack and placed into an Eppendorf tube. The supernatant was removed, leaving the WBC pellet. EBTA was added to prevent aggregation of the WBC pellet, and then RPMI was also added to maintain the viability of the cells. The cell was counted using a hemocytometer, and the WBC was diluted to obtain the count 50,000 microliters. Anti-leukemia assay. Absorbance of the leukemic blood samples treated with vincristine and absorbance of the normal blood samples treated with phosphate buffering saline were used as the positive and negative control respectively. Cells proceeded into a 96-well microtiter plate and incubated for 36.5 for 24 hours. After 24 hours, 10 microliters of the stock solution was added and then incubated for 4 hours. After 4 hours, 100 microliters of the isopropanol alcohol was also added and then incubated for 10 to 15 minutes. After 10 to 15 minutes, the absorbance was read at 550 nanometers wavelength and was compared to the positive and negative control respectively. Results. The researchers characterized Ulva lactica as to color, yellow-green, odor, rotten egg, texture, smooth or silky, length 5 to 10 centimeters. Figure 1 shows a normal blood film with occasional lymphocytes ranging from 20 to 25% per 100 WBC. Then figure 2 shows a acute lymphocytic leukemia blood film showing abundant lymphocytes and increasing number of lymphoblasts. Graph 1 presents the comparison of the average absorbance of normal blood samples treated with PBS, extract, and vincristine. This shows that the phosphate buffered saline has the highest absorbance, while the vincristine has the lowest absorbance. Graph 2 presents the comparison of the average absorbance of leukemic blood samples. This shows that PBS also has a higher absorbance, and vincristine has the lowest absorbance. Statistically, the researchers used t-test. The t-value obtained was 0.33, making it lesser than 2.353 the tabular value. Hence, the null hypothesis is not projected. For our conclusion, the absorbance of the samples is directly proportional to the amount of cells present. And the results revealed that the normal blood samples treated with Ulvan has a higher average absorbance than the samples treated with vincristine, and has comparative absorbance than the samples treated with phosphate buffered saline. The leukemic blood samples treated with Ulvan also has a higher average absorbance compared to the leukemic blood samples treated with vincristine, but has a lower average absorbance compared to the samples treated with phosphate buffered saline. This shows that vincristine affects both the normal blood and the leukemic blood, while the Ulvan only affects the leukemic blood samples. The study also revealed that there is no significant difference between the average absorbance of the leukemic blood samples treated with the extract of Ulva lactica and those treated with vincristine. The researchers therefore conclude that Ulva lactica is a potential anti-leukemic agent against cancer cells that is low cost and have minimum or no side effects. That's all. Thank you for listening.